first of all, I would like to thank all the HFSP committee and the organization for providing us this funding grant that was obtained only in 2024. That's my first HFSP grant. Probably it's going to be my last one because I'm now, as you can see, not really young. But it also means that you can also apply for HFSP grant even when you are out of age. So youngness is not only necessary. So this grant is about the microbial communication and in the phycosphere, and I would like to explain some of the details and how it came from. And the idea is that when we start about thinking about light, and especially at the microbe scale, and especially in the ocean, light is not evenly distributed. You have first the problem with light being absorbed by the ocean. You also have the fact that we saw it today, fluctuation of the light that can lead to day-night alternation, cloud coming around, and also you have, if you are in the sea, the wave that will change the light intensity on the bottom. You also have other light sources beside solar light. You have bioluminescence, which is quite common in the depth, but you can also find some light around the vents at the bottom of the floor. So this light comes from the Earth. So when you are a microbe, what is light for? And it's just like for the butterfly. The light can be used to observe prey or to hide from prey. It can be also used to get some food as a photosynthetic organism or as a phototrophic organisms. So we started in this field like that by wondering about, in fact, this came from a discussion with this guy here, Idan Tuval in the Balear Islands. So I think it was one of the best places, I think, to have your lab in Mallorca. So he cannot be French, of course. So this guy has been working, he's a biophysicist, and he's been working on diatoms, and especially the behavior of groups of diatoms. So he's biophysicist into active matter. And he discovered that if you look at the way these algae, it's algae broad sense, are moving in the liquid, in fact, they do not have random motions. When they are all together, they tend to synchronize themselves, and the synchronization seems to be due to the fluorescence of the chlorophyll. So basically, they respond to blue light in terms of absorption, but they synchronize their motion with the red light that is re-emitted by the chlorophyll. And so this is, for him, a strong research axis now. So he's collaborating with people that you already heard about, like Angela Falciator in Paris. This idea came to see if we could generalize this to the phycosphere. So for people like me that didn't know what it was, let's say 2 years ago, the phycosphere is basically the consortium of cells that surrounds the algae in the sea. And they are living on the surface of the algae. They are using the extracellular matrix as a food. And usually they return, it's a kind of cooperation, they return nitrogen-based compounds like ammonia or amino acids, and also growth factors and vitamins. And the interaction between these two is something complicated. It can be positive, but it can be also negative. They can try to kill each other by changing the composition of the chemical signals. And this is the core action of Jean-Baptiste Renard, who was in Sydney when we started to launch the program. And now this guy is back to Europe. He's in Bremen, so moving to Australia to Bremen is awesome. Okay, possible. So what about other eukaryotes? In fact, if you look at what was interesting to us, and especially to me, is the fact that diatoms are not only an algae, you have also this silica shell around, and this silica shell is called the frustule, is known to have optical properties. So basically, when you apply light on the frustule, light will be tamisé, will be sieved by the frustule because of the photonic properties of the silica construction. And this may also happen on the coccolithophores, which are calcium carbonate algae. So the idea would be to see if other cells could also be implied in this kind of phenomenon. And that's why we asked Glenn Wheeler from Plymouth, who is also a specialist on coccolithophores and dinoflagellates, to work with us. And now we are arriving at the dream that we proposed to HFSP, and just to give you the solution, HFSP send us the message. We don't believe in it, but we will fund you. So, okay, so now we have to do it. So the 3 questions were, what do these cells interact through light together? And what would be the form of communication between the cells? Are they exchanging light, for instance, the bioluminescence with, or the fluorescence and the light sensing functions of the bacteria? And is there a feedback? I mean, if the light is better, do the bacteria produce more chemical sensors, something like that? What kind of chemical exchanges? And of course, one key question is also, how do bacteria sense the surrounding light at their level? Again, it's very interesting to know that we are going to work at the micron scale, I mean, the intimate connection between the cells. So the first thing that was done by Australia is that they were looking for the bacterial photoreceptors, and you have several of them, so I'm sorry for the blue square has moved a little bit. Basically, you have 3 big classes of chromophores, some that are sensing blue light, that is typically the love domain, so it's flavin, flavoproteins. You have the opsins, which are sensing a very large spectrum of light, and there are the bacterial phytochromes, which are sensing red and far red light. And we know that the most interesting would be red and blue light. So we started by first blue light compound. And the model that was selected by the people from Sydney is Alteromonas macleodi, which is a quite common bacterium found in the ocean. And it is always found associated with algae, and it's quite sensitive to blue light. So that was also a good point to start with blue sensing bacteria. So the setup that is right now run in Spain, so if you go to Palma, you may see this. So it's basically it's a microscope, and it's designed to host a macrofluidic cell in which you can put the bacteria in some kind of quiet environment. So it could be, you control the oxygen, you control the evaporation, and they are quiet, and they will be in a very small, narrow, almost 2D area. You see 167 micrometer thick, so there is not much room for them. And then you can shine light on them and see how they move, if they move continuously or not. And that's what the first result they got. So it's brand new. Here they apply light and they look at the motility, so it's called photokinesis. That means the cells are moving randomly, but if they change of mobility, then the images, frame to frame, will be more and more similar because they tend to stay frozen. So of course the image looks very much alike. So the highest you are, the frozen are the bacteria, the more frozen they are. And you see when you apply the blue light, the bacteria tend to freeze. And then when you switch the light off, then the bacteria return to their original motion. So here we have a proof that bacteria could sense blue light, and these bacteria are the ones that may participate to the phycosphere. The next is to go to phototaxi. So it's again, this is the Australian group, which are trying to find a way to move to see that the bacteria are thriving with the light or not, and if they are producing or active in terms of metabolism. So they use this very simple resazurin test, which is proving that resazurin is something that can be oxidized easily by the bacteria. So if the bacteria are really metabolically active, then they will change the color of the medium. And you see that if you apply the blue light, the bacteria will tend to move away from the blue light and stay where they can, let's say, reduce the amount of light received. And the next step will be now to see, so it's not very clear, you have a red square. This is a region of interest that will be screened in a channel. So we'll try to see if the bacteria are moving upside from back to fro in the channel. So now for the red light, so the bacteriophytochrome again was selected by Sydney, and we started to explore the abundance of the genes of these pigments among all the oceans, and especially according to the depth. And we've seen that in deep sea, bacteriophytochromes, although there is not much light, red light at this level, they are quite present. So basically, this bacteriophytochrome could be used to sense chlorophyll fluorescence. And it's true that if you look at the spectral overlap between the fluorescence and the phytochromes. You see, I have highlighted in green the region where the fluorescence of the chlorophyll will overlap with the bacteriophytochromes. You can imagine that there could be a transfer of information between the fluorescence, the chlorophyll from the algae, and the bacteriophytochromes from the bacteria. So here, what they did is to mutate Escherichia coli and include the genes that are coding for bacteriophytochromes to produce and to enhance the production of a red-emitting protein. So basically, when if the construction is active, then the gene will be expressed, and you will see this red light coming out. And that's what they've done, so it's very successful. The only problem now we have is that the bacteriophytochrome, as you can see, has a blue, I don't know if you see it. No, anyway. There is a band at 400 nanometers, which is basically the blue light. So you can wonder if the bacteriophytochrome will also respond to blue light, despite that everything is set for red light only. And so the cherry on the cake was to establish a modification of the local climate around the cells. And that's where I come as a chemist, because my job is not biology, it's chemistry. And we propose to use these kind of tiny crystals. There is somebody in the room that knows them very well. And these crystals are called UCNP, upconverted nanoparticles, and they are made of, it's a crystalline structure in which you introduce rare earth. So if you know all the tension between Europe and China about the rare earth, well, we are part of it, but at a gentle level. And basically, it's a cocktail of ions, and if you shine light in the infrared light, in the infrared region, this crystal can produce blue light, UV light, red light, and green light. It depends on the composition. And these are almost like narrow lines, so it's almost monochromatic each time. And the thing that was extremely spectacular, that these crystals have been injected into the eyes of a mouse, and the mouse now is sensing infrared light by the tiny amount of green light produced by the crystals. And this is that was the fundamental idea for the HFSP project, is that you can amplify the light that is produced by these crystals using living matter. So basically, here the retina acts as an amplifier for the amount of light that is really produced by the crystals. And so the question is, would be any metabolic loop able to amplify this light if the cell is sensing the light? Anyway, we're not at this level, but we have to prepare these. So just to show you what this looks like. So here you have, c'est comme ça. You have the scheme of nanocrystals. So the nanocrystals are this grayish object you see at the bottom. And they are 50 nanometers, and it's quite complicated in terms of architecture. It's what's called core shell architecture, with layers containing the elements that you want to play with. And we have chosen an excitation at 808 nanometer using a special rare earth called neodymium, because at 808 nanometer, this infrared light is poorly absorbed by water, so you do not expect to cook your algae. You don't want to make a soup. I'm French, I know what is a soup, but I don't want to make soup with diatoms. That's not my key. So you can then apply the red light and this infrared light, and according to the sensitizer you use, you can produce then pure red light or blue light. And then you have the spectrum, you see. It's a very sharp line spectrum, and it matches quite very well with the porphyrins. And so we are at this stage now, which is to incorporate these crystals into beacons. And these beacons will be beads, so you have several shapes here, from 1 microns up to 200 microns. We generated them by simply either polymerizing the polymer around the beads, or by precipitating polymers with the nanocrystals all together. And our idea now is the next step will be to make this beautiful disco dance floor for bacteria, where you will have traps that look like V, in which we will stuck the beacons, and then stop the flow after the bacteria are in, and then apply the infrared light and see if the bacteria will be dancing around the beacons. So we'll have a mimic of what could be called local optical climate. Voila, I think I'm arrived at the end of the film. I would like first to acknowledge, of course, the funding agency, the HFSP. I'm really indebted, and this gives me a really kick for that. And then the four labs involved. So Idan Tuval in Spain, which is really the inspirator of this project, Glenn Wheeler in the UK, Jean-Baptiste Renard, who has a very good talent to writing grants, is very sharp. I'm reading that he was really smoothing everything. And you see, all the postdoc we were able to hire thanks to the HFSP money, and especially the 2 people in Toulouse here, and that produce the nanoparticles that you've seen. And I would like to thank you all for your attention again. Thanks to the HFSP organization.